All right, this mini lecture is on lipids. Just some characteristics and terminology. They call lipids hydrocarbons because lipids are mainly composed of carbon and hydrogen atoms. Another thing you should know about lipids is that they are insoluble in water. They do not dissolve in water, and that's because lipids are non-polar and water is polar. Okay. There are about 3 functions of lipids. They store their energy in their bonds between the 2 carbons and between a carbon and a hydrogen. Those bonds have energy. They are structural molecules because lipids are a part of a cell membrane in animals. And they also form fat in animal bodies, which serves as warmth, thermal insulation. Okay, a typical lipid is called a triglyceride. Tri means 3, and then you'll see where the glyceride comes from for the glycerol. So, what does the 3 represent? The 3 in the triglyceride is representing fatty acids. There are 3 fatty acids in a typical lipid. Fatty acids are nonpolar, so they are not attracted to water, and they are hydrocarbons, which means they are mainly composed of hydrogen and carbon. That's a big long chain, and then that chain is attached to a polar carboxyl group, which is one of the functional groups you should have learned in the first section, or carboxylic acid, which is abbreviated COOH, but technically that is short for a carbon double bonded to an oxygen and then a hydroxyl group attached to that. That is called a carboxylic acid, or a carboxyl group. So these 3 fatty acids are also bonded to a glycerol molecule, and that's where the glyceride part of the word comes from. A glycerol, it ends in OL, that means it's an alcohol, and it has 3 hydroxyl groups. Remember, hydroxyl are the OH groups. To make a triglyceride, so to make a typical lipid, you would have to have 3 condensation reactions. So we'll see that in the next slide. Okay, so this picture is showing the making of a triglyceride, or the making of a lipid. You can see we have 3 fatty acid molecules. A fatty acid is made up of carbon and hydrogen, and you can see it's a big, long chain, okay, of single bonds. And then at the tip of the fatty acid, you have your carboxyl group. And you can see the OH is there. So there are 3 of those, okay? And then what we're going to do is we're going to bond that to a glycerol. A glycerol is an alcohol, which means it has OH in it. And you can see here, there's your OH groups. Okay, so we're going to do 3 condensation reactions where we're going to remove water. So we're going to remove water right here, 1, 2, 3 times, and that is going to form the bond. You can see that with the arrow. So we are removing 3 molecules of water to bond this together, and when we bond that together, now we have a triglyceride. This bond right here where the arrow is called an ester linkage. That's just the name of the bond, kind of like the glycosidic linkage with the carbohydrates. All right, a little more detail on your fatty acids. Fatty acids can be short, they're typically long, and they can look a little bit different. And the difference is between saturated and unsaturated. Fatty acids can be saturated. If they are saturated, that means they are full of carbons and hydrogens. They are saturated with hydrogens. That means they have the maximum number of hydrogens that can be allowed, all single bonds. If the fatty acid is unsaturated, then that means there are double bonds involved. And so when you have a double bond, and you'll see this in a diagram in a minute, it'll cause a kink or a bend in the structure. Okay, so here's a diagram of palmitic acid. This is a fatty acid, okay? And we need to figure out, is this a saturated fatty acid or is this an unsaturated fatty acid? When you are looking at the molecule, you see all these single bonds between the carbons, okay? Those single bonds form a nice linear molecule. It's nice and straight, and then you can see your carboxyl group at the tip. But because all of these bonds are single, that means this is a saturated fatty acid. It has the most hydrogens that a fatty acid can have. All right, this is linoleic acid. It's still a fatty acid, okay? And you can see it still has the structure of the carbon, the hydrocarbon chain, and your carboxyl group up at the top. However, if you are looking at between the carbons, you notice highlighted in yellow, right here and right here, there are double bonds. The double bonds allow this molecule to not have the maximum number of hydrogens available. Because of that double bond, you can see there's single hydrogens attached instead of two hydrogens attached, if you compare it to the one we just did in palmitic. So what these double bonds do is it causes a molecule to have this bend. And so you can see fatty acids that are, this is unsaturated, are bent due to this double bond. Another term that you should be familiar with is amphipathic. Fatty acids are amphipathic because they have a hydrophilic end and a hydrophobic end. So that term amphipathic means that they have both hydrophilic and hydrophobic parts in the same molecule, which is what a fatty acid does have. Another type of lipid is the phospholipid. You might have heard of phospholipid before. A phospholipid is a type of fat that you would find in a cell membrane. And so it's different than a triglyceride because it has two fatty acids, not three fatty acids like a triglyceride has. It has two fatty acids, and then it has the glycerol, so that's the same, but it also has a phosphate compound attached to it. This phosphate group has a negative charge, and so that is going to make this molecule hydrophilic. So the end of it will be hydrophilic. So let's look at the diagram. Okay, don't worry about this name. This is a phospholipid. Okay? Both of these pictures are phospholipids. On the left, you can see the chemical structure of it. You have two fatty acid chains, as so. You can see the hydrocarbons. Sometimes they will abbreviate that or shorten it and not show you all the carbons and hydrogens at each of those corners. And then you got your carboxyl group. Okay, it already did the condensation reactions and bonded to the glycerol, so you have your fat there. However, this is a phospholipid, so you also have a phosphate group. Notice all of the oxygens there, and you have a choline group, which is a nitrogen in there. So see these charges of pluses and minuses. That makes this part of the molecule polar, attracted to electronegative and attracted to other polar molecules. And then you have this hydrocarbon chain down here that is non-polar. So this is amphipathic because it has a hydrophilic end with those charges, and it has a hydrophobic end here at the tail. So if I put this phospholipid, for example, in water, the polar heads, which have the positive and the negative, are going to be attracted to water. Because remember, water, right here, is the oxygen's electronegative. It's going to pull that negative charge toward the oxygen's nucleus, and these hydrogens end up having this partial positive charge. So this positive right here on this polar head, which is from the choline group, is going to be attracted to the negative on the oxygen. And this negative, which is on the phosphate group, will be attracted to the hydrogens in water. So all of these polar heads get nice and tight with the water on this side and this side of the molecule. But all these hydrophobic fatty acid tails in the middle, these fatty acids, do not like water. They're not attracted to it, and they actually repel it. So what happens in a phospholipid is that all of the polar part of the molecule goes on the outside, and all the fatty acid tails stay on the inside, and it sometimes can form a circle. All right, and then our last example of a lipid is a steroid. An example of steroid is cholesterol or testosterone, hormones. These will have this type of structure to be familiar with. It still has a fatty acid chain that's kind of like branched off of these rings. So it sort of looks like a carbohydrate because it's got this ring structure, but it's not. It is a steroid or a lipid because of this fatty acid chain that's attached to it. Usually a typical, or always I should say, a steroid will have four rings, and that's how you would typically recognize it as a steroid.